So today we're going to talk about the differences between C3 and C4 plants. Now in case of a C3 plant, the primary carbon dioxide acceptor is a 5 carbon compound. So it's a 5 carbon compound called ribulose bisphosphate, which in the presence of an enzyme called Rubisco is going to convert into an unstable 6 carbon compound, which will then split itself into 2 molecules of 3-phosphoglycerate. Okay, whereas in case of C4 plants, these are plants with a special type of anatomy called the Kranz anatomy, correct? So the leaf has a special type of anatomy. So here the primary carbon dioxide acceptor happens to be a 4 carbon compound, which is it's a 3 carbon compound, which is PEP carboxylase, and it is going to combine with carbon dioxide in the presence of this enzyme to give a 4 carbon compound, which is oxaloacetic acid, right? So this is the difference with respect to carboxylation. Now in carboxylation itself, carboxylation happens only once in case of C3 plants, whereas carboxylation happens twice in C4 plants. Okay, so where does it happen twice? Once in the mesophyll cell. So this is in the mesophyll cell. So both cases in C3 and C4 plants, the carboxylation happens in the mesophyll cells. But an additional carboxylation step happens in C4 plants in the bundle sheath cell. Okay, so in the bundle sheath cell, there is going to be presence of Rubisco, and this Rubisco will continuously combine with carbon dioxide. Where does that carbon dioxide come from? The oxaloacetic acid migrates from the mesophyll cell into the bundle sheath cell and undergoes decarboxylation, releases a molecule of carbon dioxide, and that combines with Rubisco to form the same product over there. Okay, so the primary difference is that carboxylation happens once in C3 plants, whereas it happens twice in C4 plants. Okay, this is the major difference. Then one more is the carboxylation or the presence of enzymes for carboxylation. Here we have only Rubisco, and the Rubisco is present in the mesophyll cells, whereas in this case mesophyll cells have PEP carboxylase, whereas bundle sheath cells have Rubisco. This is one more difference. Okay, then with respect to anatomy, there is no special anatomy in case of C3 plants. It is just normal leaf anatomy with bundle sheath cells, but the carboxylation is not happening in bundle sheath cells, whereas in this case we see a special type of anatomy called Kranz anatomy. What does that mean? The bundle sheath cells are quite thick walled and larger in size, and they are arranged in a ring around the vascular bundle. So this is the vascular bundle, and the bundle sheath cells are large in size and thick walled and arranged without any intercellular spaces, and this gives the appearance of a wreath, and hence it is called Kranz anatomy. And these bundle sheath cells happen to have agranule chloroplasts. Agranule chloroplast, which means they do not have grana, rather very few grana are present. So Kranz anatomy is a special kind of anatomy which enables efficient carbon dioxide fixation. And one more reason Why it is efficient in its carbon dioxide fixation is that C3 plants show a process called photorespiration, where rubisco, because it has active sites for binding with both carbon dioxide as well as oxygen, it has higher chances of binding with oxygen to form two molecules of phosphoglycerate and phosphoglycolate. So it forms phosphoglycolate, which is a 2-carbon compound, and this process is not going to produce anything useful. Rather, it is going to make use of ATP, and it is not synthesizing anything useful like sugars, okay, and it is also releasing carbon dioxide, whereas this photorespiration process is not observed in C4 plants because it has a very efficient carbon dioxide fixation method where the amount of carbon dioxide is ensured, or the presence of carbon dioxide is ensured at the enzyme site. Okay, so the primary carbon dioxide acceptor is PEP carboxylase, so there is no chance for oxygen binding at all. And even in case of bundle sheath cell, rubisco has no chance of binding with oxygen because a continuous supply of carbon dioxide is ensured at the enzyme site. So hence C4 plants are considered to be photosynthetically more efficient than C3 plants. Is this clear? So more about this we will talk later. Thank you.